Gomez. I'm a postdoc in the LSAMAT lab in UCSF, and I'm going to talk to you about quantifying feedback control in biomolecular systems. So the importance of feedback control in biomolecular systems has long been recognized. Particularly, feedback control is necessary for both homeostasis and biochemical adaptation, which are ubiquitous phenomena in biological systems. Now, in general, any system displaying feedback control can be described as in this diagram, where you have an output of interest that represents a measurement of a process to be controlled, and then the controller senses the process state and sends a corrective signal accordingly. Now, a crucial step to be able to both better understand as well as design effective feedback control in biomolecular systems is to be able to evaluate how good a particular control system is. So far, most of the analysis rely on the adaptation error with respect to some ideal or optimal output defined as the set point. This concept is inherited from control theory and engineering, where in most cases, the set point can be clearly determined from the equations describing the system. But biological systems tend to be significantly more complicated, with high nonlinearities, emergent properties, and biophysical saturation, making not only determining the set point complicated, but questioning the utility of the concept as a whole. So instead of jumping into the discussion of the relevance of set points in biology, we decided to develop a novel metric that allows for the evaluation of a feedback control efficiency in rejecting a disturbance, not only regardless of the biochemical complexity, but embracing it and fully taking it into account. And the concept behind the proposed metric is actually really simple, and for the same reason, generalizable. For any feedback control systems, by conceptualizing it as in this diagram, we can determine this crucial link, the feedback information from the process state to the controller. And we actually don't care if these boxes overlap or not, always that we can identify this link. And this link is important because by breaking it, we can build, and while introducing a new molecule that mimics the input feedback, we can build a locally analogous system without feedback, such that not only the output of these two systems will be the same for a given condition, but actually the concentration, the steady states of all the molecules involved will be exactly the same between the two systems. And this is really powerful because it allows it effectively maintains all the intrinsic biochemical properties of the system. Whatever regulatory function is in saturation will stay in saturation. Whatever nonlinear regime will be exactly the same between the two systems, with the only difference that when a perturbation occurs, the non-feedback system cannot transmit a corrective signal anymore. Then, by directly comparing these two systems under the same perturbation and the same conditions, we can effectively isolate and measure the feedback contribution from all the other particularities of the system. And this is how we do it. So for a feedback control system of interest and a particular condition rho, we have our locally analogous non-feedback system. Then we define our metric simply as by having a small perturbation around here, we divide the change in the output of the system of interest over the change in the output for the locally analogous non-feedback system. And the form, doing this in a logarithmic scale. So the form of this metric has a huge advantage, and that is that it is well-bounded in the regime we care about. It is equal to 1 when you effectively have no control. In other words, you have exactly the same output with and without the feedback. And is equal to 0 if and only if the perturbation has no effect on the system only in the presence of the feedback, which is known as perfect control. Then we can do, check a range of different conditions, and we can produce this nice and informative curve that tells you not only when your particular control system is more powerful or efficient, but also how much, as well as the range of values where you expect your control will be working. So to give you like a really, really quick taste of how powerful this simple metric is, I'm going to mention 3 possible implementations. First, we can use it to better characterize a control system. So even in the case of a well-known system, as it is the antithetic feedback, that in its mathematical abstraction is shown to provide perfect control regardless of the complexity of the process, our metric highlights right away that the small details of the biomolecular implementation can actually result in important and dramatic effects on the controller performance. On the same way, given that the metric is so easy to interpret, we can computationally optimize it to find the best possible design for a particular process and control system. And finally, by providing a unifying framework, we can also use it to compare distinct motifs and find the strengths and weaknesses of all these in a wide range of conditions and processes. And that's it. I will be happy to chat more if you're interested. Here is my contact information, and thank you for listening.
My name is Brian Davila. I'm a postdoc in Robert Blaylock's lab at UCSF. Thanks to the organizers for an opportunity to share our work. So we are interested in coordination between proliferation and differentiation, which is crucial for development. So I'm going to be... That big green button, okay. So we're interested in the coordination between proliferation and differentiation. And so we all know the cell cycle is regulated at a number of different levels, so one of which is transcriptional control. So you're looking at a schematic of how that plays out here. So you can see that there are certain genes that are actually preferentially expressed before transitions during the cell cycle. So look at cyclin E that is preferentially expressed before G1S transit as an example. So I want to make a couple related points. The first is that this is extremely prevalent in the genome. So depending on the cell type and the species, 2 to 20% of the entire genome is phasically expressed. The second point is that a lot of these genes are involved in progression through the cell cycle itself, but many others are involved in different processes, including our interest in differentiation. The last point I wanted to make is that this is not the case for all cell types. So this is data taken from the original INDROP paper. You can see a typical example on the left. So this is what you see, the big, let's look at this. Okay, there we go. A big red cluster of genes that we expect to be co-expressed in G2M show a nice correlation. And their expression is anti-correlated with genes that are expressed in G1. So you don't see that pattern in pluripotent mouse embryonic stem cells. So our interest is in looking at these pluripotent cells as they're specified into lineages. And our question is, when does the transition from this expression pattern to this one occur? And how is it regulated? So I told you before, I just told you a second ago, that a lot of the genes that are phasically expressed are involved in progression through the cell cycle. So if that's true, that would suggest that when their regulation changes, maybe we'd see a difference in the way the cells actually progress through the cell cycle. So I just looked at different stages of a mouse's development, dissociated the embryos into single cells, and asked when the DNA content profile started changing. And so I saw that it started changing after 7.5 in a mouse's development. And so we looked at that stage and tested our hypothesis. And we did that by sorting cells, individual cells, into different phases and then doing differential expression. And when we did that, what we saw is a dramatic increase. So each row is a gene. The green ones are significantly differentially expressed between G1 and G2M. And so what you can see is there's a dramatic increase in the amount of differential expression between 8.5 and 9.5. And so this does not relate to de novo expression. The genes that are phasic at 9.5 are expressed at 7.5. It's just a change in the regulation. So you see an increase in the fold change between the genes, these genes at 9.5 relative to earlier stages. So what regulates this change? So for that, we went back to earlier work from the lab. So Robert's lab has characterized 2 clusters of microRNAs that share a seed sequence. So these clusters of microRNAs suppress the G1S checkpoint. And so in pluripotent cells, they actually expedite the cell cycle. So they cause the ES cells to cycle very quickly, and they cause them to transit from G1 to S without actually stopping at that checkpoint. So our guess was that these might be involved, and so my first question was, are they expressed at relevant levels in this transition? And so what I found is that they were actually the most abundant family in the entire embryo at 7.5, and they declined sharply between 8.5 and 9.5. So then I moved to a knockout and asked, was there actually a change in the amount of cell cycle regulated expression in the knockouts relative to the controls? And what you can see here is that there's a dramatic increase in the amount of cell cycle regulated expression in the MIR302 knockouts relative to the controls. The 290 cluster, so the other cluster that shares the seed, is not actually expressed at this stage. And so the targets that are actually phasically expressed, so it's not the targets that become phasically expressed. Our model is that it's the G1S checkpoint being established. So we thought, okay, what members of the G1S checkpoint are upregulated at this stage? And we were specifically interested in ones that were actually 302 targets. So we found one that was a great candidate, P27. So we looked in P27. We expected the opposite. We saw the opposite. So you can see in controls that there's a lot of phasic expression. And when we actually get rid of P27, we see this disruption of the establishment of phasic expression. So the last thing I haven't told you is that we already know that from earlier work that in MIR302 knockouts, you get premature differentiation. So our question was, okay, if that starts happening at E9, and we've done that through characterization, so staining, we see early neurons, Was this actually a secondary consequence? So is the early phasic expression just a secondary consequence of differentiation, or is it upstream? And so to ask that question, we did it in single cell analysis, so both these mutants in single cell analysis. We saw that all the lineages were affected in P27, and in 302 we saw different lineages being affected at different times, but I did an experiment where I took the Gotkins-Marioni atlas. So this is an atlas that covers every 6 hours from 6.5 to 8.5 in mouse's development. And I lined up our control and mutant data. And what you can see here, I don't think this is a phenotype, but I want to make the point that we don't see any difference in the differentiation status of the knockouts, which are shown in orange relative to the controls. But we do see by a variety of different metrics that these cells have early evidence of phasic expression. So with that, our model is that the 302 cluster is promoting proliferation by suppressing the G1S checkpoint and timing differentiation in the embryos. Thank you. Sorry. Cheers.
Okay, I'm Shen Wang. I'm a postdoc in Russ Altman Lab at Stanford. So today I'm going to introduce OnClass, a tool for single-cell annotation based on the cell ontology. So this is the setting of our problem. We want to annotate single-cell cell type based on the gene expression profile. And here we have some cells. Most of them are annotated, and remaining ones are not annotated. And we try to know the cell type for those cells based on their gene expression. And this can be modeled by a machine learning model, for example, a multi-label classifier. However, those models cannot annotate for unseen cell type. So you see the green one, the purple ones here. So those cell types that only appear in the test data, but never appear in the training data. So when you use a multi-label classifier, you will incorrectly annotate those cells as either orange or blue, but not in green or purple, because they never appear in the training data, and you don't know how their gene expression profile looks like. And this is the question we want to solve in this project, trying to use OnClass. So before I introduce OnClass, this is an illustration of how we do it. Use an example of classifying unseen animal image to see how we classify unseen cell type based on the cell ontology. For example, we have a very good ontology of gray cats. So we know we have never seen those 6 different cats before, but we know how they are similar to each other. We know that lion is similar to leopard, tiger is similar to cat, but cat is dissimilar to lion and leopard. So we have never seen those 6 different animals before. And now we have some training data. We have training data of lion, we have training data of jaguar, we have training data of cat. So we know how those 3 animals look like, but we still don't know the remaining 3. And now I have a new image, which should be a leopard, but because we have never seen leopard before, so we cannot correctly classify this image. And what we do is actually to use this ontology to infer that this image is actually a leopard. How we do that? So we found that although it's not belonging to lion, jaguar, or cat, but this image is actually more similar to lion and jaguar, but dissimilar to cat. So it should be in a position in this ontology tree that is similar or close to lion and jaguar and pretty far away from cat. So it should be here as a leopard. This is how we annotate unseen animal. So we use the same strategy to annotate unseen cell type in single-cell data set. And the key here is to have this ontology, a good ontology of label space. And what we have is the cell ontology. So we have a cell ontology. It's a curated data source of different cell types, more than 2,300 cell types, from general cell type to specific cell types. And we use the same strategy. When we have some seen cell types, so those cell types appear in the training data, we can do query classification. And then when we want to annotate single cells, you'll see that those many cells are not annotated. And for example, if we want to annotate this cell, we found that it's pretty far away from the orange and blue and pink, so we think it might be a novel cell type. And we found that because it's more similar, more close to orange and blue, but pretty far away from pink. So it seems that it should be a cell type on this cell ontology tree that is also close to orange and blue, but far away from pink. And we can see that, we can figure out that this must be in this cell type. And this is how we annotate unseen cell type based on the gene expression and based on the cell ontology tree. And we do experiment on the tabular Murray's data set. We found that our method can accurately annotate cell type, especially for unseen cell type, especially for unseen cell type this group in comparison to existing approach. And here is how we can annotate cell type. There are 9 different unseen cell type, and our annotation is very accurate, more than 18% accuracy comparing to the ground truth. And none of those 9 cell type, we have seen it in the training data. And our method can also be used to identify marker genes. So we can identify marker genes for both cell types that we see in the training data, and also even for cell types that we never see in the training data, we can also find marker genes. And it is very accurate by comparing to a marker gene curated from the literature. And by using those marker genes to classify single cells, so we no longer use any supervised model, just simply use a marker gene. We found that for cell types that with very few cells, we have a comparable performance with human curation. And for cell types with more cells, our method can even have a better performance with human curated marker genes. And we do a transfer annotation to a large data set collection of 26 different single-cell data sets. And we found that we can do good prediction for both cell types that are in training data and cell types we never see in the training data. And this is how we do this single-cell annotation based on cell ontology. We have data code available online and also our manuscript on bioarchive right now. And I want to thank my co-author, Angela, Jim, and Ross here. Thank you.
Thank you for having me. My name is Erin Street. I'm a professor of bioengineering here at UC Berkeley and a BioHub investigator. And I want to talk to you about a really exciting collaboration with Nir Yosef and Ellen Roby over in MCB at UC Berkeley. And a bunch of our students who have been working very hard. Whoops, I'm going the wrong way already. On this data set. So much of the cell atlas to date is composed of large single-cell RNA sequencing data sets. However, much of the literature about cell types on the single-cell level is from flow cytometry, where we can identify cell types with a handful of markers of surface proteins. And so we've been recently using the CiteSeq technology, cellular indexing of transcriptomes and epitopes, which was developed by, God, I'm pressing the wrong button here, at the New York Genome Center by the Satija Lab and Peter Smibert. And what CiteSeq essentially does is it combines high-throughput single-cell RNA sequencing with highly multiplexed analysis of surface proteins. And the secret sauce is this panel of handcrafted antibodies, each which are connected to a specific barcode that has a poly-A tail, such that these barcodes look kind of like mRNA molecules. And so the strategy is to incubate your cells with this panel of antibodies, and then wash away all the antibodies that don't bind. And then you perform your favorite droplet RNA sequencing platform, which has these barcoded beads which can capture any polyadenylated nucleic acids. And so by putting both cells and beads into these droplets, you can make huge libraries and essentially count the number of antibodies from this panel for each cell and, of course, the RNA molecules. And so what you're left with is this massive data set with tens of thousands of cells and thousands of transcripts and potentially hundreds of proteins, depending on how big your panel was. And the challenging thing about analyzing this data set is while both of these matrices are over-dispersed, the protein data and the RNA-seq data is very different in its nature. The RNA-seq data is sparse, and there's a lot of zeros in this matrix. And the protein data, however, has a very large background because you get signal from antibodies that weren't necessarily bound to your protein because of nonspecific binding or ambient antibodies. And so we need a way of analyzing this sort of joint system collectively so we can perform our downstream cell atlas tasks. And so what we've been using is an evolution of the SCVI platform that was developed by Roman Lopez in Nir Yosef's lab, and we call it Total VI. And this is a fantastic collaboration that's between Adam Gayoso, Zoe Steyer, and Roman Lopez. And the brief idea is that we use Total VI to learn a representation of this joint system and then encode these count matrices into a lower dimensional latent space. And we can perform all of our denoising and batch correction in this latent space and then sample from this latent space to generate a continuous smooth count matrices where we can map all of our cell types in a reduced dimensional space. And tomorrow, Ramon's going to have a poster and a lightning talk about this, so you can ask him a lot more questions at the poster session. And so we've been using this system to study this algorithm rather, to study a really interesting process that takes place in the thymus, where we develop sort of a balance between CD4 cells and CD8 cells, or rather helper T cells and killer T cells. And so in the thymus, these double positive CD8, CD4 cells kind of go down this sort of branching trajectory and determine whether or not they're going to turn into CD4 or CD8 cells. And this decision-making process is kind of unknown. We have a lot of surface proteins that can annotate cells along this pipeline, but we don't really know those upstream events that determine how the immune system balances between killer T and helper T cells. And this balance is extremely important because we want to have an immune system that's primed and ready to respond to infections, but not hyperactive and leading to autoimmune diseases. And so we've been trying to use total VI and CiteSeq to understand this branching point here. So just a quick snippet of our data. We're using a collection of transgenic mice and wild-type mice, and sequencing a lot of cells from mice that only have CD4 cells and CD8 cells. And if you don't correct for the batches and the different genotypes and the different animals, you get a lot of different clusters. But when we use total VI, we can sort of harmonize all this data into a single latent space, and we can really nicely see this branching trajectory. And just a little a view of what this looks like. If you look in both the protein space and the RNA space, you can see that up here on the top right, we have double positive CD4 and CD8 cells, and we can see this really nice branching into CD4, both in the protein and in the RNA channel, and CD8, both in the protein and the RNA channel. And so we can use this sort of harmonized data set to generate hypotheses about the transcriptional events that happen sort of upstream of this branching. So more to come. Thank you for your attention.